Hi, my name's Kyle Johnson. Welcome to my Flash Talk. I'm a biomedical engineering PhD student at Georgia Tech and Emory University. I'll be presenting work done with Adam Willits and my advisor, Chris Rosell. So let's talk about ways to study neural activity. The most basic thing you can do is passive observation. Let's formalize this activity as the variable A. It could be firing rate, it could be something derived from it, like band power. And normally we want to relate it to some downstream variable, which we'll call B. Now, if all you're doing is passively observing, the best you can do is correlation. If you use a more active approach, like stimulation, you can say something about causation. Now, we can use even more active approaches of closed loop control to conclude even more. So one flavor of closed loop control is we refer to as reactive control, where you're delivering some predefined stimulus in response to discrete events in A. This could be used for rejecting sharp wave ripples or seizures, for example. Now, the other kind is feedback control. This is used to drive the system to a target. Michael Bolus, a former student of our collaborator Garrett Stanley, has used this to clamp firing rates of mouse somatosensory cortex in vivo, for example. But it's not totally clear at first glance what advantages this gives you over open loop control. So let's talk about that. Let's say we have a two-dimensional A, so A1 and A2. We want to study activity at the baseline and at some target with elevated A1. But let's say our baseline looks like this. You see the problem? The system spends a lot of time away from the center. There's a lot of variability. With open loop stimulation, the best thing we can do is move that variability over, but our conditions aren't very separable in our explanatory space A, and chances are they won't be in our downstream space B either. We could collect more data, which averages out that variability and makes our estimates more precise, but we can't always do that. Collecting more data costs time and subjects and money. We could also just change our target, right? Use stronger stimulation. But what if that puts us outside of a normal operating range? At that point, we're compromising our question. We could be bumping into homeostatic plasticity, for example. Strong stimulation could be harming your tissue as well. So this is where feedback control shines. By driving the system to your targets, clamping it, so to speak, now we can hit them with much less variability. Our conditions are much more distinguishable. Feedback control could be useful for things like this. Decoupling variables, you might want to increase gamma power without increasing theta, for example. State space trajectories, which have been really received a lot of attention for power in describing motor cortex or prefrontal cortex. Also for circuit identification problems, if you think of clamping the activity at a node, it is equivalent to severing the input functionally, because the inputs to that node are no longer propagated downstream. So it could be useful for circuit identification. Adam Willits is studying this extensively, so keep an eye out for a paper from him. Now, the problem is with closed loop control is it also has its costs in terms of complexity. It's not easy to take your signals in real time, compute the control signal, update your controller, your stimulus on the timescale of milliseconds, which is why we think in silico preparation is really important. First, in a simulation, you can get some idea if closed loop control is even necessary or helpful for the questions you're asking. If it is, then for any given experiment, you still have a lot of choices to make. How and where to stimulate, how and where to record, what algorithms to use. Depending on the complexity of your model, you might want to get a head start on your model fitting using simulated data, especially important if you have hard time constraints in a live animal experiment, for example. You can also prepare for things you might not know perfectly going into an experiment, like option expression. You can make your experiment robust to things you aren't sure about. So this is why we're developing a Python package called cleosim, built around the Brian simulator. Brian is flexible, easy to use, scalable. We're making an optogenetics module, easy to inject into existing Brian models, an electrode module as well to capture spiking, approximate LFP. Also, the processing loop interface is important. It allows you to model whether your control delay is 1 millisecond, 10 milliseconds, which could make a big difference. We imagine people could use cleosim for prototyping a specific experiment, finding the ideal configuration for your needs, developing control algorithms for use broadly, use a set of different models as a sort of test suite, or maybe a set of hypothetical models and you're trying to find the best configuration to adjudicate between them. In conclusion, I hope I've been able to convey some of the potential of closed loop control methods as a technique in neuroscience. Keep an eye out for cleosim. We plan to release it in a few months. And if you're an experimentalist, please let us know if this has resonated with you. We look forward to applying these ideas in practice. Thank you very much.